Hello guys, chapter 9 is fairly a little bit long, so I'm going to break it down to two parts. It's about 90 slides, so I'm going to break it down to maybe two half-an-hour sessions. I think that's better, and talk about things a little bit slower. Hopefully you get it. I'm sure you're going to get it. Hopefully you don't have to go ahead and watch it over and again. Now, there's a lot of information in there, and I'll let you know what I want. What are the stuff that I expect you to know from this chapter. Let's move on. Okay, what is flow of energy in living things? We talked about this a little bit in the previous chapter. So energy flows in an ecosystem as sunlight and leaves as heat. And photosynthesis is an endergonic reaction generate O2 and organic molecules which are used in cellular respiration. And then cells use chemical energy stored in organic molecules to generate ATP, which powers work. As I said, if I eat this, some of it, all of it, whatever, doesn't mean anything. This inside of my body turns into ATP, and ATP is allowing me to talk, to snap my fingers, and to be with you guys. But anyhow, so having said that, this is pretty much what happens with the sunlight. It goes to the plants, and plants have chloroplasts. We will talk about it later on. Inside of chloroplast is chlorophyll, which we will talk about all of this in chapter 10. And then with the help of sunlight and plants, and of course there's oxygen involved and so on and so forth, they release oxygen. Sorry about that. They release oxygen, but they take up carbon dioxide. Plants release oxygen. When oxygen is released, mitochondria inside of our cells, they give us ATP, a good amount of ATP. But we will not talk about mitochondria in this one. We'll talk about it in the next set of slides, okay? So cellular respiration, mitochondria, photosynthesis, chloroplast. And as you know, we talked about endosymbiotic evolution of endosymbiotic. These two possibly were bacteria at one time, and they were engulfed by cells, and then of course they're benefiting the cells. Okay. What are catabolic pathways yield by oxidizing organic molecules? As you know, catabolism, it means breaking down. Anabolism, anabolic steroids. You've heard of anabolic steroids. They build up muscles. When you take anabolic steroids, you inject them. Catabolic, hopefully you won't do that. Catabolic pathway release stored energy by breaking down complex molecules. We talked about that. The breakdown of organic molecules is exergonic. We talked about that. Fermentation, which you probably have heard about it in your life before, it means when molecules break down, sugar molecules pretty much break down without oxygen. So if you want to make wine or beer or anything like that, that is a fermentation. And you should not allow oxygen in your apparatus, okay? Aerobic respiration, on the other hand, another name for fermentation is an in front of the word aerobic, okay? But the other one, opposite of anaerobic, is aerobic. It means oxygen is needed. So, and anaerobic respiration is similar to aerobic respiration, except there are some, but consumes oxygen, and that's the difference, mainly oxygen. Okay. So how catabolic pathways and production of ATP are related. The breakdown of organic molecules is exergonic. We talked about that. Fermentation, partial degradation of sugar molecules to pyruvate, or we'll talk about that toward the end. We'll talk about fermentation way toward the end. But aerobic respiration is consumption of the glucose molecules that yields a lot of ATP. Anaerobic respiration is similar aerobic respiration, but it releases small amount. Cellular respiration, I talked about that. And then also carbohydrates, proteins and fats, they can break down into fuel for our body as well. It's just not carbohydrates. But this is a glucose molecule right here. With the help of oxygen, it breaks down to carbon dioxide, and that's what you exhale. When you exhale carbon dioxide, that is the end product of eating a lot of this, okay? And of course, it releases energy, ATP, which we are going to get into soon. Before we go on, in chemistry, you all had chemistry 3A. You learned about oxidation reduction. So quickly, they call it redox. Reduction, oxidation, redox. The abbreviation for reduction oxidation is called redox. Okay, and not the acronyms, the mnemonics I have for it is oil-rich. Oil-rich, which is oxidation, it loses electrons. Oxidation, lose, oil, oxidation, lose, right? And put I in there. Oxidation loses electrons. Reduction gains electrons, okay? And reg, right here. Oxidation, reduction, gain electron. Great. Easy to remember. For the rest of your life. You're going to take organic chemistry, you're going to take Chem 1A, I don't know. Easy to remember. Oil rate. Oxidation loses, reduction gains. Okay, the transfer of electrons during chemical reactions release energy stores in organic molecules. This released energy is ultimately used to synthesize ATP, which you're going to see that. Okay, chemical reaction, principles of redox. Chemical reactions that transfer electrons between reactant are called oxidation reductions reactions or redox reactions. In oxidation, a substance loses electrons. Remember, oil is oxidized. In a reduction, it gains electrons, and it's called reduced. The amount of a positive charge is reduced. Right here, for example, you studied this at the beginning of semester, sodium chloride, salt. Sodium becomes sodium cation. You remember that? So it's oxidized. And chlorine, it becomes an anion, and it is reduced. Okay? Because sodium is giving off electron, and chlorine, it gains electron. Reduction, gain. Remember, rate. Oil, lose. Okay? So sodium is losing electron. Go back to your first exam material. Okay? So simple example. This is ionic bond. You remember that sodium and chloride are bound together. It's ionically. Okay, what happens? Sodium loses electron and chlorine gains electron. Anyhow, think about these things. So again, he's talking about what happens here. I prefer the sodium chloride. The electron's donor is called a reducing agent. So sodium is donating its electron, transferring its electron. Since it's oxidized, is being oxidized, and we call him the agent. It's not CIA agent, it's called reducing agent. Okay, and the chlorine which accepts electron, we call him oxidizing agent. It's being reduced, but the agent is oxidizing agent. I hope I'm making some sense. Oh, God, I hope so. Okay. During cellular respiration, the fuel such as glucose is oxidized and O2 is reduced. So let's look at it. Glucose, right here, during cellular respiration with the help of oxygen, Glucose in a cellular respiration is fuel such as glucose is oxidized. So if it is glucose being oxidized, then it is a reducing agent. Okay, and oxygen is reduced, right? So in this case, right here, carbon dioxide is formed, and carbon dioxide is being reduced, and of course forms water and energy. Okay. What is the stepwise energy harvested via NAD? I think as time goes on, and you guys contemplate about these things and study them, it will click in a little bit at a time. Just hang on. What is stepwise energy harvested via NAD, the electron transport chain? We'll talk about right now. He's just giving you the abbreviation, but NAD stands for nicotinamide dinucleotide. We'll talk about that here in just a minute. In cellular respiration, glucose and other organic molecules are broken down in a series of steps. Electrons from organic molecules are usually first transferred to NAD, an coenzyme, and as an electron acceptors, NAD functions as an oxidizing agent during cellular respirations. Yeah, of course, yes, we'll talk about that. Each NADH is the reducing form of NAD and represents stored energy that is trapped to synthesize ATP, which we'll talk about that. Right here, they're talking about apple. Your textbook's nice. Apple. I talks about the apple. I'll bring you chocolate, ginger. Right here is the structure of NAD. Oh, here we go. The structure of NAD. So NAD, maybe you should know that is made, as I said, nicotinamide. Right here, the name of that molecule, nicotinamide. And then you have a ribose, and you have 2-phosphate attached to it, and so on and so forth. This is a nitrogenous base. What was it? Anyhow. And then again, another ribose molecule. So when you add the hydrogens right here, the electrons, you give it to them, then the rest of molecule exists here also. The rest is here. Don't think nicotinamide does not have it anymore. It becomes NADH. So the only difference between this NADH and NAD is that hydrogen right here. Okay. Reduction of NAD, and this is oxidation of NADH. Oxidation of NADH, it loses electron. Remember that oil. Reduction, reg, gain, electron. Which electron is regaining? That hydrogen right there. Okay. I hope it makes sense. Here's NAD, and then so on and so forth. We talked about it. Here's a portion of the NAD that dehydrogenase is the name of the enzyme that makes this possible. The OH, the H right here, of course, there is another H right here. One of them attached to this, right here, so you have it. But the main thing is here. What I would like to know is the name of the enzyme dehydrogenase. Okay, NAD passes the electrons to the electron transport chain. Yes, we'll talk about the electron transport chain later on. Unlike an uncontrolled reaction, the electron transport chain passes electrons to a series of steps instead of an explosive reaction. It will not happen. It's not explosive. And then oxygen pulls electrons down the chain in an energy yielding tumble, which we'll see all of this. The energy yields is used to be generated. Right here, when an explosion occurs like here, it's a lot of waste of energy. But when energy goes through steps, you make ATP, steps by step, you make ATP. That's not a waste. So the stages of cellular respiration, a harvesting of energy from glucose has 3 stages. Yes, we'll talk about this. After this, I explained this, these two for next lecture, but let's talk about glycolysis and we stop. Breakdown of glucose molecule into 2 molecules of pyruvate, and glycolysis, colored in the rest of series, he's showing you your textbook saying glycolysis in light blue, pyruvate is in orange, and oxidative phosphorylation, electron transport chain is in purple. Electron via NAD, and glycolysis, when glucose molecule becomes pyruvate, it is called the definition of complete definition of glycolysis is when 1 molecule of glucose becomes 2 molecules of pyruvate. And glucose has 6 carbon, pyruvate has 3 carbon. Where it happens in cytoplasm. And of course, it releases some energy. It releases some ATP. It's not much. It's not enough. Okay? It's not a lot of ATP for all of the actions, all of the activities we do. And then the pyruvate goes to mitochondria. In the mitochondria, it goes through the acetyl-CoA becomes acetyl-CoA, and acetyl-CoA goes through citric acid cycle. They're showing you the orange color here. The blue is the fermentation or glycolysis, and then the orange is pyruvate oxidation, and of course the citric acid cycle is right there, part of this, the orange, and then oxidative phosphorylation, which is release a lot of ATP, some ATP, a lot of ATP right there. We'll talk about it. Let's see, I hope it's not too long in this video.
As this mountain biker heads up the trail, the breakfast he ate this morning is being burned to power his bike ride.
I'm going to go ahead, stop it, and you guys watch it on your own. He has a lot of introductory, which we already talked about, all of that stuff. Sorry about that. I'm going to move on. But please watch it on your own. You comprehend it a little bit better. The process that generates most of the ATP is called oxidative phosphorylation because it powers redox, remember, reduction, oxidation. Reaction, oxidative phosphorylation accounts for 90% of ATP generated in cellular respiration. The very last one, remember that? There are 3 of them. The very last one is 90%. The smaller amount, a smaller amount of ATP is formed during glycolysis, and of course the citric acid cycle, substrate level phosphorylation. For each molecule of glucose degrades to carbon dioxide and water. By respiration, the cell makes up 32 ATP. Right here, you already know that. I'm not going to talk about it, about the enzymes. This molecule, the substrate has phosphate, it gives it to ADP. We already talked about that, it forms ATP. So that is coming up in the next few slides. Okay, so the very first one of the 3 stages, glycolysis harvests chemical energy by oxidizing glucose. So glucose is being oxidized to pyruvate. In glycolysis, sugar splitting breaks down glucose into molecules of pyruvate. Glycolysis occurs in cytoplasm and has 2 major phases: energy investing phase and energy paying off phase. So what he's saying, if you want to break down a molecule in biology, anywhere in biology, when you want to break a molecule down, you have to spend some energy. Okay, and then when that molecule breaks down, eventually at the end, it releases more energy than eventually you, at the beginning, you break down the molecule. Glycolysis occurs whether or not O2 is present. Glycolysis does not need oxygen. Okay, right here. So let's go over. I'm going to talk about this today, and then we pause, take a break, and you come back and listen. And you, the next video is over these steps. But right now, I just want to go over glycolysis and then stop the video. So what happens? Glucose becomes a molecule called glucose diphosphate. It's not. Yeah. Okay. Glucose 6-phosphate. Sorry. Glucose becomes glucose 6-phosphate as 1 ATP you need to become 1 ADP. So what happens, it attaches to carbon number 6 right here. That's what they call it, glucose 6-phosphate. Then glucose 6-phosphate, by the presence of that enzyme, phosphoglucoisomerase, of course, this enzyme hexokinase convert glucose to glucose 6-phosphate. And then phosphoglucoisomerase convert glucose 6-phosphate to fructose 6-phosphate. Okay, and then another ATP is needed for 1 molecule of glucose. That's all I'm talking about. 1 molecule of glucose. This candy that I eat, it has thousands and thousands and thousands of one of them of glucose. Okay, I'm just talking about 1 glucose molecule. So what happens when 1 glucose another fructose 6-phosphate, you need another ATP. That one goes and attached to carbon number 1. Oh, sorry, carbon number 1. So then they call this fructose 1,6-phosphate. When they say 6-phosphate, it means the phosphate attached to carbon number 6. You remember at the beginning of semester, I numbered them for you. Carbon number 1, right here, carbon number 2, carbon number 3, carbon number 4, carbon number 5, carbon number 6. Okay, you remember that at the beginning of semester, I went over that. Then fructose 1,6-phosphate breaks down to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, dihydroxyacetone phosphate. Okay, this molecule is different than this molecule. They're isomers, right? Okay. And that's what this one was trying, this picture is trying to give you, this slide is trying to give you an overall of what's happening. Yes, when glucose breaks down to 2-pyruvate, at the beginning, you need 2 ATP. But at the end, it will give you 4 ATP, and also it gives you 2 NADH. Okay, so but I went over the steps. It has 10 steps. I went over 5 of them. Conversion of glucose to pyruvate to 2-pyruvate, 2 of them. Glucose has 6 carbon, already said that. Pyruvate has 3 carbon. Okay, it takes 10 steps. 10 steps. I hope you can see my 10 fingers. The first 5 steps are explained here. Step number 1, step number 2, step number 3, step number 4, step number 5. Then he's going over each one of them in your textbook. So, and then step number 6, eventually the enzymes, in the previous steps, each step has an enzyme. Triose phosphate dehydrogenase, okay, convert glyceraldehyde 3-phosphate to 1,3-biphospho, okay, biphosphoglycerate, and 3-phosphoglycerate becomes 2-phosphoglycerate, and 2-phosphoglycerate becomes phosphoenol pyruvate, and then eventually becomes pyruvate. And you get 2 ATP here and 2 ATP here, so you got 4 ATP. So why? You guys can see in the background, they draw 2 molecules of this for you. So there are 2 1,3-biphospoglycerate. 1 glucose molecule eventually becomes 2 of this molecule, 2 of that molecule. You see the background? You see it right there, 1, 1, there are 2 of them, 1 and 1. And eventually, at the end, you have 2 pyruvates. Okay, I hope I'm making some sense. So since you have 2 pyruvates, you are converting this chemical reaction right here, converted to that chemical reactions, you're converting 2 ADP to 2 ATP here in that step, and you're converting 2 ADP to 2 ATP in that step. So you generated 4 ATP. you needed 2 ATP right here. 1, 2 ATP is needed to break down one glucose molecule. One glucose molecule needs to break down to 2 glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. 2 ATP needed, one glucose molecule, 2 ATP needed. Then at the end, each one of those molecules become 2 pyruvate, but it generate 2 ATP. The great details of this, your textbook prevented. It is in a biochemistry textbook, if you guys want to study it. You open up any biochemistry textbook, you know the details of 10 steps. You should not know each step or the name of the enzymes. I do not expect you to know that, but I expect you to know there are 10 steps involved. Conversion of one glucose molecule to 2 molecules of pyruvate. The process is called glycolysis. Okay, oxygen is not needed. Look at these steps. Go look up the textbook. There is no oxygen needed. It happens in cytoplasm. You invest 2, and at the end you get 4 ATP. You also make 2 NADH, which is important. Not now. You will see it. Not now. I will talk about it. So these are the things I would like you to know about glycolysis. It happens in cytoplasm. You make 2 NADH. The gross, the term gross, make sure you know what that means because I use it. Gross is 4 ATP and net is 2 ATP. Why? Because you invested 2 ATP. This reaction invested 2 ATP for you. At the end, you got 4 ATP. So how many ATPs you're ahead? 2. Okay. So the gross, make sure you know those terms for exams. Gross is 4 ATP and net is 2 ATP. So I think energy pays off phase. Yes. And glycolysis. What happens after pyruvate? I'm gonna stop here. And then we'll talk about this portion in next lab. I don't want you guys to get sleepy. I don't want to get tired. So let me stop